Hello, everyone, and greetings from Ann Arbor, Michigan. I'm really delighted to be giving this talk to you on the way that we use data science in my group to study what we call assembly engineering, which is understanding how to engineer new materials. So first, let's start by defining what do we mean by assembly. So self-assembly is a process in nature by which objects organize under their own forces. These objects could be atoms, molecules, macromolecules like proteins, DNA, nanoparticles, which is what we're going to talk about today. And under their own forces means that there's no individual instructions for these objects to rearrange from one structure to another structure. There's no external field telling them what to do. They figure it out on their own by obeying the laws of thermodynamics and statistical mechanics. And self-assembly is a ubiquitous process. There are many, many instances of this in nature, and it is a fundamental way in which materials also can be made. So proteins, for example, self-assemble into all sorts of structures like viruses or into lipid membranes. And cell membranes. These are all examples where the individual protein molecules self-organize into an ordered structure under the right thermodynamic conditions. So a type of self-assembly that everyone is familiar with is crystallization. What happens when you take water and you put it in the freezer and it turns into ice? Well, what's happening there is that the water molecules, which are originally disordered and in a liquid state, are suddenly cold, too cold for the liquid to be stable. And instead, an ordered solid is the stable phase. And so the water molecules rearrange themselves into an ordered structure, into a crystal. There are many, many examples of crystals in nature where you can mix and match various atomic elements to make interesting structures. So some simple examples that we're all familiar with are things like an amethyst, gemstone, or table salt. And the example of table salt on the right is really among the simplest ordered arrangement of atoms you can have. It's a very simple repeat unit, if you will. But in fact, there's a lot of examples in nature of very complex crystal structures with many, many atoms in the repeat unit of the crystal. And in fact, there's materials called intermetallics, which are mixtures of different metallic elements like aluminum and chromium and vanadium, mixed in different kinds of stoichiometries or proportions, can self-assemble into structures that are so complex that they have tens of thousands of atoms in their unit cell or repeat unit. And so when we say that these structures are highly complex, we mean they have this large unit cell or a large repeat unit. And you can see how complicated they are just by looking at some of these images. Now, in my research group, we are not so concerned with self-assembly and crystallization of atomic systems and metal alloys, but we're very focused on what we call soft matter, where our building blocks, instead of being atoms or simple molecules, are larger. They're things like proteins, DNA, virus capsids, and nanoparticles like on the bottom. Gold nanoshell or quantum dots are example of nanoparticles that can be made in the lab. And the length scales on which that describe our building blocks are between, say, 1 nanometer and 1,000 nanometers. That's the range in which building blocks, just like atoms and molecules, will obey the laws of statistical thermodynamics, and they can self-assemble into structures just like atoms do. One of the most exciting breakthroughs, I would say, in the field of soft matter and in nanoscience and nanotechnology is the ability that the community now has to use nanoparticles as atoms, as building blocks. By making nanoparticles anywhere on this, between a nanometer and a few hundred, say, nanometers, and then using different kinds of organic molecules as bonds between these particles, between these building blocks. And it's possible to mix and match together even different shapes of particles and different compositions of particles. What's exciting about using nanoparticles as your building blocks is that now we can make materials that have emergent and collective properties for all kinds of applications that mix and match properties of materials that you might not ordinarily have in a standard material. And it can have applications in photonics and catalysis and biomaterials, high energy composites. And the types of structures that are possible by using nanoparticles as atoms is very, very rich. Sometimes these ligands are small molecules, like small surfactants that have an attractive interaction with ligands, small surfactants that are stuck to nanoparticle, to a different nanoparticle. DNA is another type of bonding element that has now been used with great success to self-assemble nanoparticles made of, say, gold or silver into structures that the nanoparticles on their own would never want to self-assemble into. What is very profound to me is the idea that nanoparticles can actually self-assemble into the same structures that atoms do. So all of those complex structures that I just showed on the previous slide can also be obtained using nanoparticles, where the nanoparticles are sitting where the atoms are in these crystal structures, but of course the size scale is bigger, right? So instead of an interatomic spacing, it's an internanoparticle spacing. But as far as where all the particles are and who their neighbors are, it's the same structure. We call that isostructural. So we are obsessed with understanding, how does such complexity arise? How does the system figure out to self-organize into different crystal structures? Why does it prefer one crystal structure over another? And how does it get there? How does it do it? We know that quantum mechanics explains a lot about bonding. Thermodynamics is important in dictating what the stable phase will be. And any crystal structure that you get must obey the laws of statistical thermodynamics. But what we don't have a theory for is understanding the microscopic pathways that lead from disorder to order. So if I have a liquid and the molecules rearrange, or the nanoparticles suspended in water, say, rearrange into a crystal, microscopically, how do they self-organize? Which particle goes where and when is something that we don't understand, right? So we don't understand, for example, how these four building blocks, exactly the way in which they come together. And if we understood how they come together, and we call those the way in which particles come together is what we call an assembly pathway. If we understood assembly pathways with a fine granularity, then we could design new materials by designing their assembly pathways. So here's an example of a computer simulation of a bunch of little nanoparticles. They're all shaped as spheres, and they interact with some kind of interaction where they can't sit on top of one another, but they have some kind of short range attraction. And they start out in the simulation in a liquid phase. And so you see little tiny patterns locally, but we don't really see any large scale patterns. But then we start to see local arrangements of the particles or atoms or whatever you want to call them in this picture that now clearly have some sort of order to them, right? We see this beautiful blue pattern emerging. So that's an example of a crystallization or self-assembly pathway that these particles underwent in undergoing a phase transformation from a liquid to an ordered solid or crystal. And it's that process that we want to understand. The title of my talk is Data Science for Assembly Engineering. By assembly engineering, what I mean is actually designing and making the right kind of building blocks that exert the right forces on one another so that we can get, under the right conditions, a target self-assembled structure with desired properties. We want to start from, here are the properties and behavior that we want our material to have. Based on that, this is the structure of the crystal that we need. And so based on that, what nanoparticles should we make and what bonding elements should we use so that when we throw these particles into a bucket of water, they will self-assemble into exactly the structure that we want. So assembly engineering, the engineering of self-assembly, is really self-assembly by design. And that's what we do. So one example of this is what's called a Clathrate colloidal crystal. This is the most complex crystal structure formed from nanoparticles that has been reported to date. And if you look at these electron microscopy images on the right, every bright spot is a little nanoparticle. And the nanoparticles are shown in that bottom figure where you see these things that look like tetrahedra, but they're actually bipyramids. They're actually imagine you take two perfect tetrahedra and you squash them, you stick them together and you squash them a little bit. That's what those particles are. Excuse me. Those particles are on a scale of about 200 nanometers on an edge. But you see the scale bar in the top right that they self-assemble into these structures that are many thousands of nanometers across. And then if you go back even farther, you see a much larger crystal structure that this is a part of. This particular crystal structure is the same structure that water molecules can make that are called clathrates. These clathrates are these cage-like, very complicated cage-like structures. This particular one has 123 particles in the unit cell, in the repeat unit. And so its basic repeat unit is extremely complicated. These nanoparticles self-assembled into this in the lab by designing the shape and the length and stickiness of DNA bonds that coat the surface. And our collaborators at Northwestern made these particles and were able to obtain these crystal structures. And we were able to design them and reproduce them in our computer simulations, in what we call molecular simulations, which allow us to very easily change the shape and change the, say, how many DNA are around the surface, or how long they are, how they bond together. And so with the simulation, this is like the best view we can get from the electron micrograph in the upper right. But if we use our computer simulations, then we can see more detail about what this structure looks like. And this is an example of what a piece of this crystal looks like. So today it is possible to make essentially any kind of nanoparticle shape out of many types of materials with great uniformity, so that all the particles are roughly the same size and the same shape. They can be decorated or functionalized with different things on their surfaces. So you can, for example, have particles that are completely coated with sticky ligands so that they will stick together with others. You can, it's possible to make particles where only half the particle is coated with sticky ligands and the other half isn't, or maybe it just has a little patch of sticky ligands. But depending on how those sticky ligands or patches are arranged on the surface of the particle and the particle shape will dictate how those things self-assemble and what kind of structures you end up getting. And so it's truly an infinite design space of possibilities. Computer simulations are the perfect tool to explore design space because we can do it faster than experiments, and we can keep certain things constant and vary other things in ways that maybe experiments can't. And in fact, simulations that we've done predict a really incredible diversity and complexity of self-assembled structures from nanoparticles simply by changing the particle shape and nothing else. And so this is an image that shows 145 different nanoparticle shapes, many of which can be made today in the lab, grouped by the kinds of structures that they self-assemble into. And so the ones in the upper right self-assemble into what's called a face-centered cubic crystal. It's the arrangement that you would have if you went into a grocery store and looked at how, say, oranges are stacked up. That stacking that they do is a face-centered cubic stacking. And that's how those particles arrange as well. Despite the fact that they all have very, very different shapes. Some of them will form a different kind of cubic packing called a body-centered cubic packing. And then some of them in this plastic crystal area will form what are called topologically close packed phases. Those are very complicated phases with very interesting and larger repeat units with, say, 15 or 20 or 52 nanoparticles in that unit cell or repeat unit. And then there's all kinds of other crystals that can form based on the shape. And one would be hard-pressed to just look at any shape on here and say, oh, I know what that will self-assemble into. If I throw a bunch of them into a bucket of water. Also, if I gave you a crystal structure and said, tell me what nanoparticle shape I should use, you'd be hard-pressed to say what that shape should be. And if you have a bunch of different shaped particles, say that can all self-assemble into structures like that Clathrate structure, which one does it the best? Which one makes the best crystal with the highest yield and the highest quality, for example? Those are the kinds of questions that we use computer simulations to try to answer. And so when we do our computer simulations, we'll either do molecular dynamics where the particles we just define our particles, their shape, their orientation, and then they have forces that they exert on each other, and we calculate F equals ma over and over and over and over a bazillion times, each time moving all the particles around. Or we can do something like I showed here, which is called a Monte Carlo simulation, where you stochastically grab particles and they randomly move around and exhibit Brownian motion, just like pollen grains in water being bombarded by water molecules. And that's, in fact, the kind of motion that occurs for nanoparticles in solutions such as water. And so when we're studying these systems, we don't know what they're going to do. We start with a shape. We don't know what they're going to make, or if they're even going to make anything. We don't know if they are going to self-assemble, at what concentration or pressure or temperature are they going to self-assemble into that crystal structure. We don't know any of that. And so we have to do a lot of simulations to hope that we start to see something self-assemble. So to study these kinds of problems requires us to do really hundreds of thousands of simulations spanning many, many particle shapes. We've done, over 50,000 different particle shapes, different kinds of interactions that are possible to imbue to a particle by changing the kinds of ligands or other type of molecules that are functionalizing the surface of the particles, and spanning a range of thermodynamic conditions like pressure and temperature. We also need to run very long trajectories sometimes because we start with a liquid or a fluid, and we hope eventually it might turn into a solid, but we don't know. And so you have to run long trajectories and wait and wait and wait until you're sure it's probably not going to self-assemble into anything. And then because we want to study microscopically this pathway, we need fine temporal granularity of trajectory data. And I should have written here, we need big systems, because since we don't know what kind of crystal structure, if anything, is going to self-assemble, we don't know what the size of the unit cell is. We don't want to influence what it would be because we have too small of a box of particles. And so we have really big systems. And so all of that together means that we generate boatloads of data on a daily basis, terabytes and terabytes of data. And so we need a way to organize that data so that we can do science with it. And the second thing is that because we don't know what's going to form, we don't know what to look for, right? So when I showed that picture before, the yellow liquid that suddenly turns into this blue crystal, I couldn't have told you what it was forming from the beginning, right? We had to first figure out what it's making, figure out when a particle locally is in an ordered structure, and then color it blue. But if you don't know what you're looking for, how do you know, how can you see it? And so that means we need relevant descriptors that will differentiate local environments and how they change over a long pathway. Local environments meaning a local liquid-like disordered environment or a local crystal environment. And sometimes, or in fact, it can be oftentimes the case that crystals will form from liquids, but first they'll form a different crystal, and then they'll form the crystal that's actually the stable phase. So we need to figure out what these relevant descriptors are, and again, we don't know what we're looking for, so we need to have some kind of unsupervised learning to help us figure out what we should be looking at, like what are the relevant physics that's going on. And so we use machine learning for that. So in the rest of this talk, I'm going to kind of split it up into these two pieces and talk about data, how we do data organization, in the hopes that it's the tool that we've developed is useful to some of you. And then we'll talk a little bit about how we use machine learning once we can organize all our data. So first, let me say that in the work that my group does, we're often asking questions, scientific questions that push the limits of what available molecular simulation software is capable of. And when that happens, we write our own codes. But we have a philosophy in the group that everything we write should be open source, that everything we write should be usable by somebody else. And we have a Unix philosophy where we want to have many small focused packages that do one thing and do it well, and then you can either use them independently or combine them together in Python scripts to form complete solutions. And so this is a kind of a map that shows many of the codes that we use regularly in the group, where the darker blue color are the codes that we've developed. Our primary code is called HUMDI Blue. That's a molecular simulation code that ran all the simulations that I just showed you. That's the one that you tell it, here's the particles and here's their interactions and here's the thermodynamic parameters, and now I want to see what they do, and produces all of the data. Freud, of course, is our analysis code. And a lot of the analysis that we do in our systems haven't been done before on these types of systems. And so there's no code that we can just grab that does it. So we write our own analysis code. And then Signiac is the data organization code that I'm gonna talk about. And all of our codes are developed open source, and they're all available at this web address. You know, when I first started as a computational scientist and then as a professor in academia with my students, it was the case that every student wrote their own code to do whatever they wanted. I did that as a graduate student. I was going to study something, I wrote a code, it did it. I didn't document it. You know, I didn't write tutorials on it. I was the only one using it. When I left the group, or when my students left the group, no one could understand what was written. No one could reuse that code. We don't do that anymore. And so we employ best practices for professional software engineering so that everything that anyone develops could be used by others. So we use Git repositories and a pull request workflow. We do very careful API design to make sure that it's intuitive to the user. We document everything religiously, and we write tutorials for all of our codes at multiple levels, so that at different levels of expertise or no expertise, you can go in and learn how to use our codes. We make sure that our codes are right by constantly performing unit tests and doing continuous integration. And we use various types of containers like Docker and Singularity that allow us to use the same code all the way from a laptop to a desktop to a cluster to a supercomputer like Summit at Oak Ridge National Lab that we use quite often, which is a leadership class machine. I think it's the most powerful supercomputer in the US for open science. So let me say a few words about the Signiac framework. This is it's a lightweight software framework that we wanted to be application agnostic, so you could use it no matter what you're doing, that helps you manage and scale your file-based workflows. And the idea that it's file-based is key. And the reason we came up with this is because even though our simulation codes and our analysis codes were being used by all the members of the group and all the members of the group contribute to it, we couldn't share data with each other. Everybody was writing their own data in their own way, in their own directories, and they would have a different naming scheme for how they would do that. And so I had a phenomenal student by the name of Carl Simon Adorf, who's now a postdoc at EPFL, who said, this is silly, we can do better. And Signiac is really his brainchild. And so now we develop it, and it's also a NumFocus affiliated project. And this is the team, that the picture, the first one there of the maintainers is Simon, also V.S. Ramasubramani, Bradley Dice, and Brandon Butler are all students in my group that have bled and sweat for this software framework to make it usable by others. And many people are using it for problems that have nothing to do with science. What Signiac allows us to do is to do research that others have coined true research. True meaning that the data is transparent, it's reproducible, it's usable by others, and it's extensible, right? We want to make it easy to be able to run hundreds of thousands of jobs with hundreds of thousands of shapes and interactions and different combinations of thermodynamic parameters, and organize them in such a way that you can come back to it a year later and find exactly what you're looking for. Or you can give this data on a shared file system to someone else, and they just have to run Signiac, and they can just search and find whatever they want to. So, give you an example. Here are two common approaches to file naming and organization in a file system. So either you come up with a very long file name, like this is NA, which refers to a particular, like say, shape with a pressure of 5 and a corresponding volume fraction of 28, and I'm going to equilibrate for this many time steps, and you just make these very long file names that are cryptic and mean nothing to anybody but you. Or you have very deeply nested directories where you group things together, but still things get nested. And what's hard about this is, let's say we're, and this happens very often, a student will come and say, well, I've simulated these particular particles that our collaborators can make, and I figured out that if they do this or that, or make it this way or that way, then they can get these interesting structures. And then I'll say, well, what would happen if you change the shape a little bit, or threw in a second shape, or looked at it with slightly shorter ligands or DNA, whatever. All kinds of things. Well, then how do you modify your file naming scheme in a way that's now consistent with the idea that now you're going to open up a whole new area of design space? So that's what Signiac allows you to do. So Signiac basically designs in jobs in a workspace, and each one of these jobs is like a state point where you would run something. It's like one of those file names would be a job. And so you run the Signiac framework, and within it you initialize these jobs, and then Signiac Flow will work with Signiac to run your simulation, analyze your data, and visualize your data. And it's all written in Python, so it's very easy to use and can be used with anything. And so, for example, you can use aggregation to operate simultaneously on multiple jobs. You can use groups to combine operations that are related to one another into a single submission, right? So you can simulate and analyze and visualize, you could throw that all on a cluster and have it come back and organize all the data for you. And you can use bundling to submit scripts that will execute multiple operations. It also, Signiac integrates into high performance computing workflows. And I mentioned before that through the use of containers and Singularity, Docker, we can easily port to any computer of interest. So what Sadiak is great for is managing file-based heterogeneous data on a local file system. For searching for data, accessing it, you could do it within Python or on the command line. Developing scalable and reproducible computational workflows, including very complex workflows where you want to run a simulation for this long and then do something and do something else and then run it through the analysis. And then if the analysis says something, then you want to go back and do this other thing. This makes it really easy to do that. It's great for rapidly prototyping workflows and analysis and integrating with existing tools and running on all these different machines. There's a lot of other things it can do. For example, you can customize the parallelization for different kinds of computer architectures and generate dashboards for viewing graphical output. So for example, Bradley Dice, a student in my group, developed Signiac dashboard that will grab from Signiac your data and quickly visualize it. And so you could search for, I could search jobs for all of the simulations that were done for a particular shape or that combined a shape with a different shape, or use these different kinds of nanoparticles, and it'll just visualize, and it pulls it all up and visualizes it for me. So Signiac has revolutionized the way that we work in my group, because now we can all share data. And the results that I'm going to show you next were where we used machine learning on data. This was data that came from many different students in the group over the last 5 or 6 or 7 years. But because we could easily find the data and know where it comes from, we could use it and for machine learning. So the website is really the place to go if you want to learn more about it. Signiac.io. We have read the docs that it's well documented, and we have a Slack channel for users where you can quickly ask questions and get responses. So if you think it might be interesting for you, I hope you will go and visit that. So we have all this data. What do we do with it? So when I say what do we do with it, again, here's an example. These are 2 different samples, say, of nanoparticles in some solution, like nanoparticles in water. They start out in a fluid phase. They're different shaped particles. They start out in a fluid phase, and then they start to organize, and we can see crystal structures forming. In the one on the right, we see that there's some local organization of particles into these orange spheres, but those are not really ordered. And then somehow they all lock into place, and then we color them red, and then you see this very complicated crystal structure that actually has 435 particles and then repeat in it. And the one on the left is, the blue and the yellow ones we color them that when they're still mobile and not yet organized, and we see that there's this crystal that's growing in this layered way. Again, these are systems where we don't know what to expect, and so we have to come up with how do we find these patterns in the data? How do we find these motifs? And so we use machine learning on a Signiac data space to study how self-assembly occurs by interrogating these self-assembly pathways. And the work that I'm going to describe now is all work that was done by my student Bradley Dice, and I also stole some of his slides that he made for his thesis defense, which he is doing later today. The idea is to develop a microscopic understanding of assembly pathways using machine learning. So we're starting off with particles, and we need to have some descriptors that tell us what the local particle environment is, so that we can distinguish one crystal structure from another, and crystal structures from a liquid, or partial crystal structures from other partial crystal structures or liquid. You could come up with a bazillion different descriptors for that. And then you get this very high dimensional set of particle descriptors that somehow you have to reduce the dimensionality of, and in the end have some low dimensional representation that tells you what are the most important descriptors. And it tells you, that turns the data into useful information. And there's a lot of different structural descriptors developed in our field to study self-assembly. Most of them are developed to distinguish like a reactant from a product. I start with this, I get that, or one local region from another, but in a static structure, not something that follows it in time. And there's a lot of examples here. Almost always they're motivated by some physical intuition or expert knowledge. Like, for example, we know we want to look at what a bunch of particles are doing around a particle of interest, because that neighborhood is what makes this motif that's part of the crystal structure. So we know what we want to look at locally. But all of the descriptors that I know of from the literature are discontinuous, meaning that a particle could move a little bit and all of a sudden you have a discontinuous change in your order parameter, which is fine if you're just looking at a snapshot and saying, what are all the environments? What do I have? But if you want to follow this in time to look at the whole self-assembly pathway, we need a continuous descriptor. And also we want scale invariance, because we want to be able to compare different types of nanoparticle or atomic systems that self-assemble into the same structure, but are different on different scales. And so what Bradley came up with is a continuous topological order parameter based on something called Minkowski structure metrics, which I won't go into, but continuous topological order parameter defines a feature space of these metrics as a continuous manifold. And the manifold topology is what reveals the pathways that are sampled by the individual particles as the system goes along this assembly pathway, and as they explore different kinds of local symmetries. The way that we reduce the data, so we come up with a scale invariant high dimensional vector of these continuous topological order parameters that are calculating all sorts of things. And then we use UMAP as a dimensionality reduction algorithm. It's similar to TSNE. It's different from, say, principal component analysis, and then it's non-linear. UMAP is the uniform manifold approximation and projection that I have a link down at the bottom to the website of those who developed it. What's interesting about UMAP is that it in mapping, in reducing the high dimensionality feature vector descriptors to something manageable, it preserves the topological structure of that high dimensional manifold, which means it preserves the local distances, which means that distances in a high dimensional feature space are preserved in the reduced dimensional space, which means that local environments that are similar to one another will be close to each other in the reduced space. UMAP is also something that gives really fast performance on GPUs using rapid CUDA ML. So again, the beauty of combining these continuous topological order parameters, which I'm not explaining the details of for lack of time, and this particular dimensionality reduction algorithm UMAP is that similar local structural environments, the local neighborhoods, the local arrangements of particles, ones that are similar to each other will be close to each other in feature space and close to each other in the UMAP embedding. So let me give you an example of what this means. Here's an example from NIST. The image data is called MNIST because it was modified from the original NIST image data. That where there's all this data embedded from 784 dimensions into 2 dimensions using UMAP. And so each one of these images represents a number, a numeral. There are 28 by 28 or 784 pixels that represent each numeral. And so one can combine, one can make a vector in 784 dimensional vector that measures the Euclidean distance between these numbers. And so on the right, this is now the UMAP embedding, the 2-dimensional UMAP embedding, where the different numbers are given different colors. What we see here is that, for example, 8, 5, and 3 are all close to each other in this plot. That proximity is real. It tells us that there are pixels, that there are features about the numbers 3, 5, and 8 that are very similar to each other. It also tells us that images 7, 9, and 4 have features that are very similar to each other. And we know they're similar to each other because they're actually close to each other in this embedding. And then there's the rest of them up here, like 1, 2, 0, and what's this one? 6, that are less similar to others. And so they are more separated. And so this gives you information, not only that there are 10 numerals, which you might get with a different kind of dimensionality reduction, but it also tells you that there's 10 numbers and some of them are similar to others. And it tells you which ones. So that's what we want to use for our study of self-assembly of nanoparticles into ordered structures. So by having these continuous variables and this topology preservation, we now have interpretable assembly pathways. And so we have particles, we compute these Minkowski structure metrics, we get this continuous topological order parameter, we reduce the dimensionality with UMAP, now we have this self-assembly pathway. So as an example, here's a final frame of a trajectory of icosahedrally shaped particles that self-assembled into a crystal that's an FCC crystal structure. That's the crystal structure, remember, that is like when you stack oranges in the supermarket. And what we also see that, so all the green ones are locally in this particular crystal environment. The red ones are associated with a slightly different crystal structure called hexagonal close pack. And then there's a couple blue ones that are actually slightly different crystal structure that you randomly get, and then white ones are locally just disordered. So this is at the end of the day, we can identify these and color them. But what the machine learning process algorithm gives us is the following. So this plot on the left, so this is tricky to explain, so I'm going to do it really quickly though. This plot on the left, there's a dot there's a point for every single particle at every single time step in a really long simulation. So there's a bazillion points all plotted on top of one another. The points that are blue are points from early time in the assembly pathway, and the ones that are yellow are what all the particles are doing, like what local environment they have. at the end of the assembly pathway after the crystal has formed. And so this shows us that the system, most of the particles are in this fluid phase, and then over time, there's a range of different crystal structures. So you see how the yellow is kind of spread out into this contiguous shape, this contiguous manifold. That means that there's a distribution of local structures that the system has at a late time. What we also can look at is it also tells us what those structures are. So we can see that it starts out as other, meaning disordered, it's nothing. And then we see that most of the points are bright green, which means that they can be characterized as FCC. But then we see this little red part, HCP, the hexagonal close pack. So it tells us, by looking on the right, it tells us how many different kinds of local environments our system sampled in going from a liquid to a crystal. And it also tells us time-wise how it did it in this kind of continuous way. And so this UMAP projection tells us about that the system is undergoing these continuous deformations. Because the embedding is a continuous manifold, and the points nearby in the UMAP embedding are also nearby in the high dimensional CTOP space, and because nearby points have similar symmetries in their local environments, just like the numbers are similar in their local environment, we can follow the assembly pathway from fluid to crystal and know how every particle's local environment changes in time along this pathway. Very quickly, I'll show you, for example, four different systems that are all starting in a fluid and crystallizing into the same exact crystal structure. They're just different kinds of systems, different kinds of particles, different kinds of interactions between them. But that shape, that's similar for all of them, tells us that the assembly pathway is similar for all of these different systems. Because they're forming the same crystal structure despite being very different kinds of systems. And it also allows us to compare going from, say, a fluid to a crystal structure, or from a crystal structure back to a fluid, or from one crystal structure to another. That's what all of these different shapes are telling us. Remember, the blue is how it starts. The brightest yellow is where it ends. But what these tell us is that all of the local environments that are sampled by the particles are somehow connected or accessible from all of the others, so that the symmetries of local environments have a lot in common. And that helps us to understand how to make new systems that will form the same kind of crystal structures, but in different ways, or different kinds of structures. And if we take all of these co-embeddings, or we take all of these UMAP embeddings and put them together in a co-embedding, we can see the whole structure of the manifold of all of the different crystal structures that can form from all of the different systems that we're looking at that start as a fluid and end as a crystal. And the fact that they make this contiguous, I don't know, looks like a manatee, tells us that you can access these different crystal structures from one another by going through different kinds of things. So this information helps us to engineer new assembly pathways. So to conclude, some of the takeaways from this talk are that opportunities to realize new materials with assembly engineering require new ways of organizing all that data. Signiac is a software framework that provides an easy-to-use free platform and facilitates the use of machine learning to discover hidden connections with very little prior information about the system. And by combining a continuous topological order parameter and a dimensionality reduction algorithm like UMAP, provides a way to analyze and interpret these assembly pathways, again, without knowing what we're looking for. And so I'll just conclude by listing the various agencies and places that have funded and supported our software development. And now I would be very happy to take your questions.
Thank you, Sharon, very much for this excellent talk. So there are a few questions that people are asking.
Great.
So the first one is, does the initial random arrangement of the particles have an effect on the resulting self-assembled structure? If so, how do you account for this in your simulations, and what steps do you take to study these divergent pathways further?
That's a great question. The individual positions of every particle at the beginning, of course, contribute as to exactly what moves where. But the beauty of statistical thermodynamics is all of that kind of random information doesn't matter. And so the pathways followed are independent of how they're randomly arranged. If they're arranged in a very different starting configuration, then that will matter. But from one random initial condition to another, the self-assembly is a robust process, and so doesn't care about that. And of course, when we do simulations, we run multiple simulations starting from different random initial configurations to test that that's the case.
I see. Okay, thank you. Another question is about your software engineering practices in Python. Mm-hmm. So, do you share resources, guidelines, what books to follow? I mean, this person is just asking, could you share these? Are these resources like Signiac? Is it already online? And I think it is already online.
Oh yes, everything's available. Everything's available. If you Google me and go to my group website, you will find all of the information. We share everything on GitHub. If you search for Glotzer Lab on GitHub, you'll also find all of our software. The Signiac software, an easy to remember website is just signiac.io.
Great. So I guess I would like to ask one question. It's very interesting that you use the MNIST data set, because in KDD, probably every nth paper uses that data set, so we're very familiar with it. And you mentioned about the UMAP projection, and that it preserves sort of distances in some sense. We also use random projection often to we take a high dimensional data set, apply sort of essentially multiply by a random matrix. Mm-hmm. And we get a low dimensional feature space. And there's a very famous mathematical result called the Johnson-Lindenstrass lemma, which actually states that it does preserve with high probability. So is this UMAP has some properties like that? Is it what is the complexity of this algorithm? Do you know?
That's a great question that I can't answer, not being an expert really on these techniques. I know that it's a relatively new algorithm that we found because we were trying to find something that would give us physical insight, right, into this feature space. But it's a great question.
So once you find these low dimensional projections, then you said you just get the pathways. What exactly does that mean? I was just curious.
So it means that we start out so some of the examples I gave where you start as a fluid, a liquid, and you end in a solid. And what we want to know is, okay, but was it that simple? You just started as a liquid and something happened and they ended a solid. What is the something that happened? And that something that happened is something that is structural, it's local, and we want to understand what exactly that is. Part of the challenge of that is that the things that happen in the middle of the pathway may have nothing to do with the final crystal, with the final product that you have. And in fact, sometimes you can create one crystal that then turns into another crystal. And so now that we have this information, we can go in more detail and figure out what exactly was the microscopic process that led this liquid to become this ordered structure. What I'm even more excited about is being able to look at many, many different substances that all start as a liquid and end as the same essentially the same structure and ask how many different types of pathways are there and how different are they from one another?
Let's see. Okay, good. Well, yeah, thank you very much, Sharon. I think it's an excellent talk, and I think this is a great. There's lots of opportunities for collaboration with data scientists and people in say material engineering, physics. So thanks again very much.
And thank you to the audience for your patience and letting me talk about the science problems that are compelling us to use some of these techniques. And I hope some of them are also useful in your own work.
Thanks a lot. Bye-bye.
Thank you.
Take care.